I am Dr. Sumana, Associate Professor, Department of Microbiology, JSS Academy of Higher Education and Research, Mysuru. The objective of the study was interesting and the curiosity to assess the presence of macrolide-lincosamide-streptogramin resistance among the lactobacillus that are isolated from various food materials and also animal-derived food, and their conjugal transferring capability to the native microbiome. The study was objectivized to trace the dissemination of antibiotic resistance between animal, human, and zoonotic microbes, which is posing a threat to global public health. Here, lactobacillus, being a reservoir of antimicrobial resistance genes, is suspected for the possibility to transfer the genes to native microbiome in both human and animals. This transfer of resistance may be due to the horizontal gene transfer, referred to as HGT, that are happening among the microbial pool, which may be considered as an important criterion. This is due to the fact that most of the lactobacillus are used as starter cultures and they have to be screened for the presence of resistance gene and also its capability to transfer to native microbiome. The second main areas focused are the indiscriminate use of antibiotics for the enhancement of growth in poultry and livestock raising, and the macrolide-lincosamide-streptogramin resistance gene that are reported in many bacteria. As the MLS resistance gene was noticed among various bacteria, the current study was designed to assess the MLS resistance transfer by horizontal gene transfer, or it is otherwise referred to as conjugal gene transfer, or it is otherwise synonymously referred to as conjugation method, from the isolates of lactobacillus that are isolated from various food materials. Ready-to-eat fermented meat foodstuffs deliver microbes to gastrointestinal tract of humans. If the animal microbe has resistance gene and capable of forming conjugation, may transfer resistance genes to gut microbiome, posing a threat towards the commensal's resistance gene acquisition and opportunistic gut microflora, which may lead to antibiotic treatment failure. Further, screening of lactic acid bacteria for the presence of resistance gene and its transferability to native microflora is of priority, as they may pose a serious menace on human health. Based on the objectives, the study was designed in such a way that it is explained in the schematic representation here, where the bacteria were screened and isolated from different food sources and were identified as Enterococcus faecalis from sheep intestine, Enterococcus hyrae from chicken intestine, Lactobacillus fermentum from fermented idli batter. The isolates were checked for general minimum inhibitory concentration, and the antibiotics like rifampicin, tetracycline, vancomycin, erythromycin and clindamycin were checked for the resistance that is posed by the bacteria that were isolated from different food sources. And the concentration of the antibiotics were increased to 3 to 4-fold to isolate the resistant bacteria. Isolated bacteria were considered as donors, and the conjugation or the transferability were checked with the recipient bacteria. Recipient bacteria used in the study is Enterococcus faecalis and Acinetobacter. Table 2 shows the bacteria that were isolated from different food sources and their resistance against the different types of antibiotics and on the selective media with the different concentrations of antibiotics. Here, the donor and the recipient strain were subjected over for the transferability or horizontal gene transfer through these three methods, that is by liquid mating, solid mating and filter mating method. In vitro conjugal transfer of macrolide-lincosamide-streptogramin resistance gene was studied by these three methods, in which the filter mating method was found supportive in assessing the horizontal gene transfer by three-folds than the other two methods, with the donor and recipient ratio being 1:1, 2:3 and 3:2 in the study. The table 3 depicts the donors list and their sensitiveness or the resistance against the tested antibiotics. Table 4 speaks of the ratio of the donor and the recipient and the combination of the donor and recipient, which was found best in 1:1 ratio among all the other recipients, and it was observed that the best was seen in 2:3 ratio among the donors. Table 5 speaks about the transfer frequency, which was found high in filter mating method than by the other two methods that was studied, and this ranged from 1.7 to 3.7 into 10 raised to 5 bacterial cells. To determine the conjugation or horizontal gene transfer from the donor to that of the recipient cell, and to determine that transferability from the molecular perspective, the transconjugants were subjected to RAPD, that is random amplified polymorphic DNA analysis, using M13 primer. This confirmed the transfer of genes from donor to that of the recipient bacterial cells. Supportive to that of the molecular studies regarding the gene transfer in the transconjugants, the transconjugants were inoculated on solid media with the antibiotic that showed the phenotypical expression or the phenotypic growth of bacteria, confirming phenotypically that conjugation or the transfer of resistance genes that has happened between donor and recipient bacteria. So figure 2 speaks about the isolates that are inoculated onto the solid media with the phenotypic expression of the growth as well as no growth, on the contrary, with the presence of the concentration of the antibiotics. Figure 3 speaks about the molecular studies that was extended to find out the presence of resistance gene transferred from the donor to that of the recipient cell with respect to the resistant gene, that is ERMB gene, and its primer that was used to check the presence of this ERMB gene, resistance gene, presence in the transconjugants, and it was found positive through the molecular studies. Similar type of presence of the other resistance gene, that is MEFA-E genes, which is also a resistant gene, was checked in this transconjugants using the primer, and the recipient exhibited the presence of that gene. The presence of MSRA-B resistance gene in the transconjugants also proved to be present in the recipient or the transconjugants, which proves the presence of these three genes in the transconjugants or the recipients, which have received them from that of the donor cells. So to summarize the work that was carried out, through the morphological and molecular studies that was performed, revealed that lactic acid bacteria is a very potential candidate to transfer the resistant determinants, that is the genes, which may intersect in the food chain. Study highlights the possibility of horizontal gene transfer by lactic acid bacteria. It is a debatable issue where lactic acid bacteria, which is exploited in food industry, which may be a potential candidate in transferring resistance or the resistance determinants or the genes, and also serving as a reservoir of such determinants. Current study emphasizes on judicious usage of antibiotics in human, veterinary, poultry, animal husbandry sectors, where lactic acid bacteria towards antibiotics bridges the development of antibiotic resistant pool, which may get transferred to commensals and opportunists, turning them to become pathogenic, leading to antibiotic treatment failure. Finally, to conclude, awareness about antibiotics and its resistance, which may magnify if not kept under check. So with this note, I would like to thank the International Research Excellence Best Paper Awards for selecting my small piece of research work on lactic acid bacteria being a very good reservoir of antibiotic resistance genes and its capability to transfer to that of the native bacteria, converting the commensals and the opportunists turning out to be the pathogens and the antibiotic treatment failure. Thank you all.